RuBisCo. For this capstone presentation, we will be discussing and analyzing the enzyme RuBisCo, which is short for ribulose 1,5-biphosphate carboxylase oxygenase. But why is RuBisCo important? The answer is simple. Without RuBisCo, we would all be dead. Life on Earth began with plants, which supply the Earth's atmosphere with the oxygen in which we breathe and need to survive. RuBisCo is present in abundance in these plants, making it the most abundant protein on Earth because it is a key enzyme in the Calvin cycle, which synthesizes sugar from carbon dioxide, giving these plants their food and energy. As the activation energy required for the Calvin cycle to occur is quite high, the reaction would only occur extremely slowly if RuBisCo was not present, or even not at all without an adequate input of environmental energy. Without RuBisCo, and thus without the Calvin cycle, any aerobic organism would cease to exist. This presentation seeks to explore the exact mechanisms by which RuBisCo is synthesized and the way in which it performs its role in producing sucrose. It will also discuss how its structure relates to its function, the strengths and weaknesses it possesses as a result of its structure, as well as the overall Calvin cycle and the molecules and processes RuBisCo influences along the way. When RuBisCo is synthesized, 16 amino acid subunits are produced, 8 of them being long and the other 8 being short. The RBCS, or small gene coder, codes a chain of approximately 120 amino acids, while the RBCL, or large gene coder, codes a chain of approximately 475 amino acids. These chains are then assembled in the chloroplasts of plants, where the enzyme stays and functions in the stroma. As mentioned in the previous slide, RuBisCo is formed of 16 subunits. These subunits form surrounding a magnesium ion, in a way similar to how hemoglobin forms around an iron ion. The active site of RuBisCo is arranged around this magnesium ion, but this active site is very large, meaning it can bond to both the carbon and the oxygen in carbon dioxide, which can result in an issue for the Calvin cycle. This is the structure of RuBisCo in plants and different types of algae, while there is also RuBisCo that can be found in some bacteria. RuBisCo that is found in plants is a larger version, while RuBisCo that is found in bacteria is only formed from 2 of these protein chains. Both of these types of RuBisCo perform its catalytic task just as well. So, why do plants build these larger complexes? The answer lies in the fact that RuBisCo performs its job in crowded conditions. By packing all these chains together into a tight complex, the protein reduces the surface that must get wet by surrounding water, which allows for more protein chains and thus more active sites to be packed into the same space, making these enzymes more efficient.
To give an overview of the Calvin cycle, where RuBisCo is most useful, the process starts with carbon dioxide molecules being transformed into phosphoglyceric acid through a process known as carboxylation. Then, through phosphorylation, ATP donates one phosphate group, becoming ADP, to the phosphoglyceric acid molecules, transforming them into diphosphoglyceric acid. The third step, reduction, involves NADPH2 molecules releasing their hydrogen atoms to become NADP, thus turning diphosphoglyceric acid molecules into glutaraldehyde phosphate. The third step, reduction, involves NADPH2 molecules releasing their hydrogen atoms to become NADP, thus turning diphosphoglyceric acid molecules into glutaraldehyde phosphate, or GALP for short. One of these GALP molecules is then released from the cycle, undergoing organic synthesis to become starch, which is then stored as an energy source. The remaining GALP molecules then undergo a series of reactions to become ribulose phosphate. Through another instance of phosphorylation, ATP molecules yet again release phosphate groups, becoming ADP, and transforming ribulose phosphate into ribulose biphosphate, thus allowing the Calvin cycle to start again. This last step between the end of one cycle and the beginning of the next is what RuBisCo catalyzes. To conclude, this presentation discussed how RuBisCo is synthesized, its structure, the strengths and weaknesses it has as a result, but most importantly, the changes in enthalpy that occur throughout the Calvin cycle. By finding the total change in enthalpy of the reaction, it is possible to then calculate the energy stored in each molecule of sugar as well. Alternatively, Hess's law enables us to use the standard enthalpy of formation for glucose, the eventual end product of the Calvin cycle. Its heat of formation is about negative 1,271 kilojoules per mole. So for every 3 moles of carbon dioxide involved in the Calvin cycle, with 6 moles of glucose being produced in the process, 7,626 kilojoules would be released. RuBisCo is necessary to the maintenance of life on Earth. It is the centerpiece of the Calvin cycle, which enables the energy captured during photosynthesis to be stored as food. Although RuBisCo is not the most efficient enzyme, the role it performs is irreplaceable. Without the presence of RuBisCo, the 10% loss in energy per food chain level would result in humankind being unable to exist. Besides, who doesn't like eating their vegetables?